Today, I wanted to report about the role of neurotrophic factors in the pathogenesis of glaucoma and to provide the results of our multi-year studies, our joint studies, where besides neurologists, ophthalmologists also participated and also biologists. Well, I will not speak too long about the role of neurotrophic factors that is very well known. They used to say that the decreased level of neurotrophins may lead to the apoptosis, triggering of retinal ganglion cell death. So neurotrophic factors are the regulatory proteins of the nervous tissue that contribute to the proliferation, differentiation, viability, maintenance, and functioning of neurons. There are several subfamilies, and this is the classification of neurotrophic factors and how they're classified. So we see the representatives of the main subfamilies before the studies, specifically NGF, then brain BDNF, then GDNF, glial cell line derived neurotrophic factor, and ciliary neurotrophic factor, which is CNTF. So for all these factors, the reasons why we selected them is very clear, because there are some previous publications that, using different glaucoma models, show us that introduction of the factor could provide neuroprotection and improve the survival of retinal ganglia cell. And specifically, it is shown here for BDNF and for the CTNF as well, in the publication of Peace and Domasini and Lee. And here I'm giving some publications for glial factor that also proves a very important neuroprotective role. Besides, neurotrophic factors are actively used in the clinical application. First of all, CNTF. This is the implant-producing CNTF by the Neurotech United States that contains genetically modified cells that could produce CNTF for quite a long time. So the implant is sutured or somehow placed in the vitreous body, in the bulb, and then the drug is already approved by FDA after phase 2 clinical trials. So it is allowed in the molecular telangiectasis of class 2, and in glaucoma there are active clinical trials on the possible use of it. So the other factor which is used in the clinical practice is just NGF, nerve growth factor. So it is approved in drops in neurotrophic keratitis, and you can appreciate the treatment which is produced commercially with a brand name, and that is quite effective. So this is a design of Stanford University. And also there are clinical trials of using drops with NGF in patients with glaucoma. So keeping in mind all these factors, to us, it was quite surprising to see that we have no information about the content of the factors in the anterior chamber, in the tear fluid. Speaking about the anterior chamber fluid, it was quite the only publication by Jodhavi et al. That did not identify the factor rates. So that was proteomic analysis of the fluids taken during cataract surgery, and they simply measure presence or absence of particular proteins. It has been shown there that there are some amounts of BDNF and NGF, but for CNTF and glial factors, there are some receptors found. So most probably they were there, but they were not yet identified properly. If we talk about the blood serum and blood plasma, there we see quite many publications, but all of them were focused on the neurological pathology. So almost the only serious publication from 2017 studied just two factors, BDNF and NGF in the blood serum. And I will talk about it later several times. And last but not least, here you see in tiers, these are own publications, our publications with co-authors and a little bit of later Iranian co-authors, also our studies in diabetic neuropathy, in case of keratoconjunctivitis. So that's it. Not many in glaucoma. In glaucoma, you see just three publications here. Let's say for B and BDNF, this is just the surface several publications. For glial factor, just one publication where they just prove the presence, but the concentration was never measured. So the objective of this clinical study was to identify and to measure four important, I would say even more important subjectively neurotrophic factors, BDNF, CNTF, NGF, and GDNF in the aqueous in the lacrimal fluid and blood serum patients with POAG. So we totally recruited 270 people, 270 eyes, each one eye, and patients were split into groups, which is a comparison group, then patients with cataract and with no glaucoma, and the main group here are patients with cataract in combination with glaucoma. Keeping in mind that we have just a little bit of fluid in the anterior chamber, we carried out the study in the two-step. First, we studied BDNF and CTNF, and then the other two markers. Here you can appreciate inclusion and exclusion criteria, which I skip. So the standard ophthalmic examination and sampling was taken just before the surgery, intra-op. Then all that was going through the ELISA analyzer. First of all, we studied the influence of age-related cataracts to the neurotrophic factors. So we measured for two factors. So we compared patients of very similar age and similar gender, where there was some cataracts, and we compared them with healthy volunteers. So both for BDNF and for CTNF, it appeared to be that no difference, neither in the TFL fluid, neither in serum. So we couldn't see the difference. And then, as I said before, we started patients with cataracts with and without glaucoma. So we checked by stages, but please appreciate we used Mills classification from 2006, where the advanced stage split in two, the advanced itself and severe advanced. So that means stage 4. It's not a terminal stage here, but severe. So that means more advanced of the severe. So what did we have at the end? So in case of CNTF, if you take all the patients, we saw the reduction in the fluid of anterior chamber and in the tear fluid. If we look at serum, then CNTF was minimal there, so we couldn't find any associations. Now, if we look at the anterior chamber fluids and tear fluid, so we saw consecutive reduction of the concentrations up to stage 4, which was very severe, and we saw the dramatic reduction. If we compare the changes in the anterior chamber and the index of the visual fields, severe, which is reflecting progression of glaucoma, then here we see some consecutive reduction of the concentration of the ciliary neurotrophic factor. And totally different picture was obtained for the brain-derived neurotrophic factor, BDNF. So here you also appreciate some reduction if you take all glaucoma patients, both in the anterior chamber fluid and in the tear fluid and also in the blood serum. Although when we checked along the stages, we were quite surprised. So it appeared to be that the maximum reduction, and in fact, not only in the fluid, but also in serum. So that means in the whole body, the maximum drop was reported in the first, very initial stage. And then we see continuous increase of the concentration that never reaches normal levels where patients have cataracts but no glaucoma. And if we appreciate visual field index, which is geometric index VFY, so we see that with progression glaucoma, with the reduction of index, we appreciate dramatic increase of neurotrophic brain factor BDNF in the fluid of the anterior chamber. This is our data, confirmed a donor study in the serum of glaucoma patients. Here you can appreciate the same. This is a column chart from his article. So we see that maximum reduction at the initial stage, which is mild and moderate, and only in advanced stages. Definitely, he didn't split them in two. So they reported the increase, which never reached the controlling parameters. And the reduction of the initial phase is also shown in the another publication from Iraq. Then nerve growth factor, NGF. So here we found no correlations, and everything looked quite similar. That means internally with cataract, with no glaucoma. Well, here we have very small samplings. That's why we put together stages 1, 2, 3, 4. Stages 1 and 2 were very similar to cataract, and stages 3 and 4 actually no differences and no dependence from the visual field index. So for all of them, so that relation was minimal. Although I have to say from the very beginning that in a donor publication that also studied that factor, we found the other results found for BDNF. So that means the reduction at the initial stages and a later increase that never reaches the normal range. I don't know why we had some discrepancy in data. Well, possibly it is in this group of patients we have the minimal number of samples, so just 25 patients with glaucoma, and possibly that was not enough. But at least we are planning to recruit some more patients and try to have more detailed assessments. In case of glial factor of BDNF, the situation was like with BDNF, with the brain neurotrophic factor. So in fact, initial and advanced stages 1 and 2, all parameters were reduced. So that means the parameters of this neurotrophic factor, they were not only in the fluid of chamber and in the serum and in the tear film, but also in the blood serum. So that means in the whole organism, we saw the reduction of GDNF content. And then it was increasing by stages 3 and 4, but in the anterior chamber fluid, it never reached normal ranges, so only in the blood serum. When we compare it with visual field index, we also appreciate that with the progression of glaucoma, we see the increase here, but in the anterior chamber fluid, the increase never reached statistical significant, but it was significant in the tear film and in the blood serum. So here we see two different types of changes of neurotrophic factors In case of CNTF, this is a reduction, consecutive reduction with glaucoma progression with minimal at stage 4. For BDNF and GDNF, and possibly even for NGF, it appears to be that, let's say, add-on is more correct in our group than possibly not. So, but at least for two factors, we can say that there is a minimal in initial stages and then increase in the advanced stages that never reaches the normal range. So these results clearly indicate the important role of the neurotrophic factors in the pathogenesis of POAG. Here are two options, and one of them I will provide as hypothesis. We all know that glaucoma and pathogenesis of glaucoma mainly related to the local disorders, for example, with ophthalmic hypertension and also decreased number of neurotrophic factors like BDNF and also in the whole organism. But this change could persist for quite a time without the onset of glaucoma. Let's recollect hypertension study. So only in 5 years, in 5 to 10%, they could confirm the developed glaucoma. So there is a thinking here that these local changes switch to glaucoma with the effect of particular factors. And as hypothesis, it could be dramatic reduction of neurotrophic factor level, possibly all of them, BDNF, GDNF, NGF, so in the whole organism, blood serum and lacrimal fluid, and also the anterior chamber fluid. So it could be whatever. It could be COVID, it could be flu, it could be, when hypertension switches to glaucoma, or maybe some other local changes also could move to glaucoma. But this is just the hypothesis, and there could be another thing. So all these changes, reduction of neurotrophic factors level, are not the reasons, but simply the sequence, the consequence of glaucoma per se. So that means they're secondary. Here you see three main publications on the data presented in this speech. So you can find them in the open access, in molecular vision, in the current eye research, and they all reflect the recent data presented here, and the latter is available online in molecular vision, so publication of this week. With that, thank you very much for your attention.